Okay. Okay. Hi, everyone. Thank you for joining our live session. So tonight we're going to be covering the first part of the three-part lecture series of enzymology taught by Dr. Ahmed Al-Jada. So in today's session, I'm taking your guys' lecture apart and focus this lecture on the most high-yield concepts and the most important foundational concepts for you guys to be able to tackle the second and the third enzymology lectures. And throughout this lecture, you'll see some of Dr. Ahmed Al-Jada's slides, just kind of copy and paste on the screen. And at other times, you'll see his slides, but with a few changes. And these slides are going to be colored in orange, just to indicate the fact that they've been adapted from the original slides, just to make the concept more simple for you guys. And the last thing I'll say before we begin is that we have 2 checkpoints throughout the session where we'll stop and take any questions you guys have. So please wait until you see a stop sign to ask questions. And at the end, we'll be covering 2 MCQs as well. And at the very end of the lecture, you guys can ask as many questions as you want. So to begin, these are the objectives taken directly from Dr. Ahmed Al-Jada's slides. So you guys can read it in your own time and won't spend too much time on it. But I've made sure that we kind of cover all of these things. So to begin, we're going to start with the terminology. The most important terminology to understand when you're talking about enzymes and enzymology is substrate, products, and the enzyme. And along with the enzyme's active site. So the substrates are your reactants. Those are the things that you're putting into the enzyme. The enzyme, you can think of it as basically like a factory. So if we were to take an example and say like, okay, maybe the substrate is like your raw material. It's, let's say, the flour, and then you have the bakery. And then the active site is a small portion within the enzyme that is specific for what you're baking. Let's say you want to make cookies, so you need an oven. The active site is your oven, okay? And so then the substrate enters the oven, and then the oven is within the bakery. And then at the end, you have your cookies, which are your products. What makes enzymes specific is their active site. So enzymes are specific to their substrate because of their active site. And again, whenever you want to remember that, just think of the analogy like you need ovens which are in bakeries for cookies, but you might need a different device for a different type of substrate to create a different type of product. And we'll talk more about this later. But basically, you can expect the specific shape and chemical environment within each active site of an enzyme to yield a different type of reaction. And we can expect the active site of an enzyme like lactase, for example, which breaks down lactose, to be different to the active site of an enzyme like sucrase. So whenever you hear a question about what determines the specificity of an enzyme, it's always the active site. Moving on with terminology, activation energy is the energy required for a reaction to occur. It's also known as the energy barrier. So we can also understand this as the energy required for reactants to transform into products. But before a reactant transforms into a product, it needs to reach the reactant's transition state, which is a certain level of energy at which the reactant can actually transform into a product. So here from Dr. Al-Jada's slide, there's a little diagram that shows A as your reactants, and then it transforms into its transition state before it can reach the point at which it can transform into a product. So this is important because enzymes utilize this idea of activation energy in a transition state, and they actually speed up this entire process by lowering the activation energy to reach a transition state. And we'll talk more about this in a minute as well. So before we get into that, the 5 characteristics of enzymes to know is that enzymes are globular proteins, which act as biological catalysts that speed up the rate of a reaction. I've also written or RNA because Dr. Al-Jada also said that sometimes you can have enzymes that aren't quite proteins, they're RNA. So you have like RNA splicing enzymes like ribozymes. Those aren't technically protein, but they do act as enzymes. They have enzymatic activity, so we include them in the classification. So that's the number 1 characteristic of an enzyme. They're usually proteins or RNA. The second one is that enzymes are catalysts. It basically means they're chemical agents that accelerate a reaction without being changed in the process. So they can be recycled, which is going to get into point number 4. The third thing is something we already talked about. Enzymes have an active site, which specific substrates bind to, what makes an enzyme selective and specific is its active site. And then number 4, enzymes are recycled. They don't get changed. They don't get consumed by the reaction. They catalyze, so they can be reused. And lastly, enzymes are typically named after the molecules that they react with, which is called their substrate, and they end with the suffix ase. So for example, lipids are broken down by the enzyme lipase, or lactose is broken down by the enzyme lactase. So if you hear the last suffix of ase, you know you're dealing with an enzyme. So how enzymes work, they do 2 things. One, how they increase substrate to product formation by lowering the energy barrier, aka the activation energy, and they increase the favorable orientation of colliding reactant molecules. So basically, this little molecule describes both of these things. So you have the substrate over here. It's just going to reach your enzyme. It's going to form an ES, enzyme substrate complex. Because the enzyme now and the substrate are linked together, we can form the product. And we'll talk about this red arrow in a bit. And then after that, you get your enzyme product complex over here, and then you get the product separate from the enzyme. The enzyme is untouched though. Otherwise, it has no remnants of the product, so it can be recycled for the next substrate to come in. And then they dissociate from each other. So that's why it can be recycled. But essentially what's happening in this arrow, this part over here, we're going to start discussing in a bit. But the first thing I want you guys to understand is lowering of the energy barrier basically allows the enzymes to have more reactions with less energy, and they increase the favorable orientation of colliding reactant molecules. Basically means they allow the substrate to come into close proximity with the enzyme in the correct proper orientation and in the correct and proper chemical environment as well. So we'll talk more about this because this is fairly important. So we can spend a little bit of time on this slide, and after this we'll have a little checkpoint. So enzymes lower the activation energy so that more reactions can occur with less energy. So this is adapted from Dr. Jada's slide as well, but I've added a few more things just to summarize it because he had 2 separate tables going on, but I kind of combined those 2 to make it a little bit more simple. But basically you can think of enzymes and the role of enzymes as you would think of cheap labor. You can equivalent energy to money. So a catalyzed reaction, which is the reaction in blue, it doesn't need that much energy for a reaction to occur, but an uncatalyzed reaction, so a reaction with an enzyme, requires more energy. So you can think of catalyzed reactions like the one in blue as like if you're outsourcing your labor to another country and you're getting things done at a cheaper price, you're getting less energy and you get the same products. So that's kind of the cool thing about enzymes, is that they allow this to happen with less energy, which means that it can happen more often. So enzymes speed up the cell's chemical reactions by lowering the free energy of activation, and it does it so much so that the reaction can occur about like 100,000 to 1,000,000 times faster than an uncatalyzed reaction. So I'll go ahead and explain this diagram to you guys a little bit now. So the x-axis explains the progress of the reaction, and the y-axis is your free energy. So as the reaction increases, as the time increases, the amount of energy over here, you can see how that increases with the uncatalyzed reaction. And the point over here, the dashed line, indicates the T point, which is the transition energy. So this is the state at which the substrate can now become the product. And as you can see over here, the free energy of activation is indicated over here. That's a lot more for the uncatalyzed reactions as compared to the catalyzed reaction. So in the catalyzed reaction, I just realized that this should be longer to show you guys what I'm saying. So for the uncatalyzed reaction, the free energy of activation actually is this part and this part, these 2, and then the free energy of activation is much less. It's just half in the catalyzed reaction, so you require less energy. So energy basically can be things like heat. So in your body temperature is at 37 degrees Celsius. You can't apply heat to your body. Therefore, enzymes are very relevant in terms of biology, because enzymes, without you having to increase your body temperature, you can lower your activation energy and you can still have these reactions form. Whereas maybe externally, without these enzymes, you would have to increase the temperature. So in the lab, if they're doing things without an enzyme, the same reaction would require more energy and more temperature. And the important thing to know is that despite the free energy of activation being more in an uncatalyzed reaction and less in a catalyzed reaction, the Gibbs free energy, the energy produced, which is the difference between the energy of the products and the energy of the reactants, remains unchanged. And you can see that over here. This energy between whatever products I've formed from my reactants is the exact same whether the reaction went through a catalyzed version or an uncatalyzed version of the reaction. So there is no effect on the delta G or the Gibbs free energy of the reaction. And the last thing to say from this table is that when you have no enzyme, you'll have fewer reactant molecules that reach the transition state. And when you do have an enzyme, you can expect more reactant molecules to reach the transition state simply because it's easier. Again, it brings you back to that cheap labor analogy. It's easier to, and you're likely to experience more of these products being made if you require less energy to make those products. So we're going to stop over here, and if you guys have any questions, I'll take them now before we move on to the models of action. I'm going to check the chat as well. What's the transition state? Okay, the transition state is the energy required for the reactant to transform into the product. So the transition state, you can think of it as like the highest amount of energy that the reactant reaches before it transforms. So I put that in T over here and highlighted it. So as you can see, the transition state for an uncatalyzed reaction is a lot higher than the transition state for a catalyzed reaction. What's the significance of delta G being 0? Does it mean reaction is at equilibrium? So delta G is not at 0 in this example. Delta G is basically the difference between the products and the reactants in terms of their energy. So whatever the delta G is, let's say it's 5, like there's a difference of 5 in the energy, that remains unchanged whether you're in an uncatalyzed or a catalyzed reaction. It's not at 0. If it were at 0, then you would assume that maybe the reaction didn't occur, or that, I'm not sure if if the reaction would have occurred or not if it was at delta 0. Isn't that the activation energy? Okay, so the transition state is a certain amount of energy that the reactant has to reach before it can transform to the product. Very similar to the activation energy. Activation energy more so kind of applies to the term of like the entire reaction, like how much the reaction requires. But the transition state refers mostly to like the reactant itself. So you can think of it as like the activation energy is the energy required for the reactant to reach its transition state. They're very similar things. Hope that makes sense. If it didn't, you guys can continue to text on the chat. But if there aren't any other questions, then we can move on. But I'll give you guys another like 10 to 20 seconds if you want. I hope that last part made sense about the transition energy or the transition state and then the activation energy. So like in this definition here, the activation energy is the energy required for your reactants to reach their transition state and transform into products. So your reactant, with its activation energy, then it reaches a transition state in which it can form a product. So if I put my activation energy here in this little arrow here, that's what's going to help me achieve my transition state. Think of it as like, I was trying to like make real life examples. Like imagine if you put like so much stress on a person and then they break down. I don't know why I chose a negative example, but think of the stress that you're putting on the person as your activation energy. And then that peak when you're like, I am this close to tears, that's your transition state. And then you reach the product of crying. Think of it like that. They're very closely linked, but the transition state mostly refers to like, so we need activation energy to reach the transition state. Yes, exactly. I think you said it much better than I could have. Thank you. You're welcome. Okay, so let's move on then to models of action. Okay, so we have the older and the newer models of action. We have the lock and key model and the induced fit model. They've recently proved, not recently, I think it's been a while, but they've proved that the induced fit model is much more kind of realistic in terms of how these things actually work. But usually in biochemistry, you're required to know and understand both. So in the lock and key model, it's similar to exactly what you would think. You have a lock, you have a key. The lock fits perfectly inside the key. It's a perfect match, and you open it, you get your products. So the enzyme's active site, according to the lock and key model, is complementary to the substrate precisely, like perfectly. And the theory of enzyme substrate interaction explains how enzymes exhibit specificity for a particular substrate. So that's the way in which scientists used to believe that, okay, this is how an active site makes an enzyme specific. It's because its shape is a perfect fit for the substrate. But then scientists did a little bit more research, and then they realized the induced fit model makes a lot more sense. So according to this model, the enzyme's active site isn't a complete rigid fit for the substrate. Instead, the active site undergoes a conformational change when it's exposed to the substrate to improve the binding. So this theory explains two more things that the lock and key model couldn't quite explain. It explains how enzymes can exhibit broad specificity. So basically, lipase is one enzyme. It can bind to a variety of different lipids. And it also explains how catalysis may occur. So the conformational change stresses bonds in the substrate, increasing its reactivity. So it just explains how basically it's the same concept, but there's a specific chemical environment within the active site that when exposed to the chemical environment of the substrate, a conformational change occurs that basically makes it fit so much better. It's like Play-Doh instead of just like a perfect metal lock and key. It's almost like the enzyme kind of conforms to the shape of the substrate more so than just being super rigid. And it makes sense, right? Because I don't think every single substrate would have a perfect rigid structure. Some might be a little bit altered or deranged, and that explains why enzymes can still work to create reactions between all types of substrates. So we do have a question. So enzymes aren't specific to just one substrate because lipase can bind to multiple lipids according to the induced fit model. I mean, yeah, not just like one substrate, but one substrate type. So you can expect like lipids to all have the same outer formula. You can expect them all to have the same like hydroxyl group at the end, general like structure. So they are specific to one type of substrate, but we're actually going to get into this in the next two slides. There are different types of enzyme specificity. So I'll wait to get back to that question actually once we get to the following slide. So just right back to the induced fit hypothesis. This explains how enzymes can react with a range of substrates of similar types. So they are similar. It's not the exact same, but here we can see how this is your enzyme hexokinase. So when it binds to the glucose substrate, you can see how it kind of closed in. It's like hugging it a little bit. So it changed its shape a little bit to induce the fit. So this is going to answer the question that one of you guys asked, hopefully. There are different types of enzyme specificity. So like we said before, enzyme specificity is determined by its active site. An enzyme selectively recognizes proper substrates over other molecules, and specificity is controlled by structure. So the unique fit of a substrate with enzymes controls the selectivity for the substrate and the product yield, as well as the chemical environment. And enzymes may recognize and catalyze a single type of substrate. This is if they have an absolute type of specificity. This is for an example of urease, which only catalyzes urea. The other example is a group of similar substrates. So this would be called group specificity, like hexokinase we just saw over here. It adds a phosphate group to hexoses, and it catalyzes one type of reaction for similar substrates. And then the last one is linkage. So this is for a particular type of bond. It catalyzes one type of reaction for a specific type of bond. An example is chymotrypsin, which catalyzes the hydrolysis of peptide protein bonds. So I'm not sure where lipase would fit into this, but it doesn't necessarily, again, have to be one particular type of substrate. It is still specific, but there are different kind of levels of specificity depending on what your body needs in whichever specific organ. Okay. Hopefully that answers your question. If it doesn't, go ahead and you can continue writing in the chat. Can you repeat? Yes. Okay. So there are different types of enzyme specificity. There's absolute, there's group, and then there's linkage. So for absolute specificity, the enzyme catalyzes one single substrate. So for example, here, let's say urease, it only catalyzes urea. Then you have group specificity, which catalyzes a similar group of substrates. So hexokinases adding a phosphate to hexoses. Hexoses are a group of 6 carbon sugars. So you can expect group specificity enzymes to be able to react with an entire group of 6 carbon sugars, whereas urea is a little bit more specific for the absolute example. Then lastly, you have linkage. So chymotrypsin catalyzing the hydrolysis of any and all types of peptide bonds, catalyze one type of reaction for a specific type of bond. So then these would be the most broad specific, or the most broad type of specificity for enzymes, because it can catalyze anything with the peptide linkage, for example, for chymotrypsin, or maybe it would be a different type of linkage for another. So this would have the most broad specificity. But in general, think of it as 3 different levels, and they seem to be increasing in the amount of specificity, or not the amount of specificity, the amount of things that they allow within their specificity to catalyze as you go down the list. I hope that made more sense. Again, if it didn't, you guys can continue reading the chat and hopefully I'll address it. Okay, now the 6 major classes of enzymes. This slide, when I saw it on the lecture, so lipase can be in group. Yeah, lipase can be in the group, exactly. Okay, when I saw the slide in your guys' lecture, I was like, whoa, what's happening? And then Dr. Chad was like, you don't need to know this, that's fine. You just need to know the general classes of each of these things, and very basic questions are going to come in your exam. You just need to understand the classes. Just kind of notice the difference in the number of reactants and products for each class. And I have a much more simplified version of this in the next slide that I'll show you. But I'm going to give you guys an example of kind of what kind of questions might come from this. He might ask you a simple reaction with 2 molecules linked together. Which type of class of enzyme would this occur in? And obviously just looking at this diagram, you're like, what? Like, I wouldn't be able to tell you that. Another example that he gave in your guys' class is 1 molecule is converted to another molecule without any other reactants. And you're supposed to know that that's isomerase. So it's really hard to tell with this. So this is the PALS way of explaining this slide. It's the same thing, but we've just removed the unnecessary information, and I'm giving you guys what you would need for your exam, inshallah. So transferase, ligase, oxidoreductase, isomerase, hydrolase, and lyase, oh my gosh, sorry, are the 6 major classes of enzymes for you guys to know. So you guys would need to know for transferase that there's a functional group, which is X, okay? Not BX, just the X. So you transfer a functional group from one substance to another. So as you can see here, it's first with B, now it's with A. And these functional groups can be things like methyl functional groups, acyl groups, amino groups, phosphate groups. And the type of enzyme would be like a transaminase or a kinase, but you don't really need to know that. All you need to know is just kind of remember this general structure, and this is the type of thing that he would like test you on mostly. And then a really important keyword for this that might come up in the question is like, if you hear the word like methyl, acyl, amino, phosphate group, the addition of like one specific group, that's basically what a transferase would be responsible for. And it's the transferring of basically this one group from this substance to this substance here. The second thing is ligase. This one is, self-explanatory. You have one substrate, you have another substrate, and then you just combine them together. It's a simple reaction with 2 molecules that are linked together. Ligase, you might remember from MOL 1, DNA ligase. It's basically just like adding 2 strands of DNA to each other and making one single strand with each other. So if, there's a question that's like, okay, you have a simple addition of one molecule to another molecule, and then you end up with just one molecule, what enzyme maybe is responsible for that reaction? It would be ligase. Then you have oxidoreductase. It's a little bit different because it involves 2 different types of reactions, but it's in the name, oxidoreductase, that an oxidation and reduction reaction is happening. So the little colon that I've added over here is indicative of your electrons, and it can go both ways because, as you know from your oxidation and reduction reactions, I always remember the mnemonic oil rig. So oxidation is loss of electrons, reduction is gain of electrons. If you're losing your electrons, then over here, it's going to be oxidation. So B is getting oxidized. If you were to go the other way around, then A would be getting oxidized, and etc. And then RIG, so the RIG would be reduction is gain of electrons. Basically, anytime that you're having some kind of transfer of electrons, you're having the involvement of hydrogen or oxygen, then you can easily think, okay, oxidoreductase is probably the enzyme that's doing things over there. And it's also pretty simple because the types of enzymes would be called oxidase, or it might be dehydrogenase, but mostly you'll hear the presence of electron transfer or oxygen transfer or hydrogen transfer. And then you have isomerase. It's basically an intramolecular rearrangement. Some change is going to happen within substrate A that's going to transfer it into substrate B. You basically are converting one molecule to another without any other reactants. So this one is pretty simple to remember. Hydrolase, basically the formation of two products from a substrate by hydrolysis, so the breakdown of water. And then essentially like your B and your C over here are going to have little parts of that water. Maybe the hydrogen will bind to your B and then the oxygen will bind to your C. But whenever you hear the term kind of hydrolysis or the addition of water to create two different substances, you know hydrolase was probably involved in that reaction. And these are enzymes like lipase, amylase, peptidase. But again, I don't think you would need to know that at this level, at least not for this lecture. You might have gotten into that in enzymology number 2. And then lastly, lyase, simply just the breakdown of one product into two different products. So A over here is just being cleaved into B and C. So I'm going to ask you guys the same questions that Dr. Ahmed asked in his lecture when he presented this slide, but I'm going to ask it with this kind of infographic over here. So if I were to ask you guys now, what enzyme would be involved in a simple reaction with two molecules getting linked together to create one molecule? Which enzyme do you think was responsible for that reaction? Okay. Yes, ligase, exactly. Okay, and now ligaments. Okay, I'm gonna pretend that was a typo. I think ligase is what you were trying to say, but ligase is the correct answer. Okay, and now the same question that he asked earlier. One molecule is converted to another molecule without any other reactants. Which enzyme was probably responsible for this? Yeah, isomerase, exactly. So inshallah, if you guys just kind of memorize this structure, you guys will be fine for the exam. He said that he'll definitely ask questions about this and that he's only going to ask it in a very simple way. He wrote, very basic questions will come, just understand the classes, and that's it. So those are your 6 classes. If there are any questions, I'll take them now, and then we'll get into the next part. Okay, I'm going to continue. If you guys have any questions, just go ahead and write it in the chat. We're going to talk really quickly about what a zymogen is. A zymogen is an inactive precursor of an enzyme. It's secreted by living cells. It gets converted in the presence of something like an acid or a kinase into its active form, which we call a proenzyme. So the purpose of having zymogens is that it allows the cell to control when and where an enzyme becomes active. So I know you guys all took GIT. In GIT, we talk about how chief cells secrete pepsinogen. Chief cells secreting pepsinogen is an example of one of your body cells secreting a zymogen. So pepsinogen is a zymogen. And then a zymogen, like pepsinogen, gets activated under the presence of a low pH, so acid, which is secreted from parietal cells in the lumen of your stomach to form pepsin. So pepsin then would be the active enzyme that breaks down amino acids in your stomach. So that's basically all that is said here. A zymogen is the inactive precursor of an enzyme, and then it has to be activated either with a acidic environment or a kinase into its active form, which then can go ahead and do the actual work of an enzyme. And again, the purpose of having all of this is just so that you can control where and when an enzyme becomes active, because you wouldn't want something breaking down proteins to be activated in a place where you don't want to break down proteins, and vice versa for pancreatic enzymes, which are also discussed here. So that's very basic concept of Zymogens. And yeah, I'm bringing this checkpoint back in case you guys have any more questions. That's kind of it for the very basic enzymology things. We're now going to start getting into enzymatic activity. So again, same thing with the chat. If you guys have any questions, we can ask them there. Okay, so for this, can you explain point B? Sure. All right, what's point B? All right, let me know what point B is, and then I can explain it. But I think I'll move on to this and then just kind of write a little explanation. And I think whenever we're done with enzymatic activity, we'll get back to that, inshallah. Okay, so for factors affecting enzymatic activity, you have the temperature, you have the pH, you have your substrate concentration, you have coenzymes, cofactors, ionic concentrations like your salt ions, and then enzyme inhibitors. I believe these will be discussed in your second enzymology lecture, but these ones were discussed in your first one. So I'm going to be spending a lot of time discussing the temperature, the pH, and the substrate concentration, because those are the three different factors that affect enzymatic activity that involve graphs. So whenever you see a graph, definitely high yield. Always, pay attention to graphs and try to understand what they're saying. But we're going to start with the temperature. Basically, a low temperature is indicated over here, a high temperature is indicated over here, optimal temperature is around over here. And as your temperature increases, the reaction velocity, otherwise known as the speed of reactions, is going to increase, because enzymes with added energy, of course, it's just going to continue to do more and more and more reactions until they reach a certain point at which it's like the optimal temperature to create reactions. And then if you heat up an enzyme too much, because an enzyme is a protein, it can do something called denature. So it gets inactivated because your enzymes are starting to denature. As more and more enzymes denature, you can expect the reaction velocity to also decrease and less reactions to occur until all enzymes are denatured, and then the reaction can no longer occur. So that's why when we have a low temperature, it's insufficient thermal energy for any type of reaction to occur. Increasing it increases the speed and motion of your enzymes and your substrate, so resulting in higher enzymatic activity. And then you reach your optimal temperature, which varies, by the way, for different types of enzymes. And then a higher temperature slowly causes the enzymatic stability to decrease. And then the hydrogen bonds are broken, different bonds inside of the protein is broken, and then you have your denaturation. For most enzymes, the optimal temperature is within your body's temperature, 35 degrees Celsius all the way to 40 degrees Celsius. But most enzymes usually are fully denatured by 70 degrees Celsius. And this graph kind of summarizes and explains all of that. Okay, so Q10 on your guys' slides basically says that there's a 10 degrees Celsius rise that, let me actually open it up for you guys and see if I can add it. Okay, so it's your temperature coefficient. So the increase in the reaction rate with a 10 degrees Celsius rise in temperature. So as your chemical reactions increase, so as the optimal temperature for an enzyme-controlled reaction will be a balance between the Q10 and denaturation. So it's basically kind of summarizes the temperature increasing by 10 degrees Celsius at each point for chemical reactions. So as the Q10 increases from like 2 to 3 to 4, you can expect the rate of the reaction to double and triple with that. It's like if you were to do it through algebra, it'd be 10x. So it would be just multiplied by 10 every single time. It's just another way of saying that the chemical reaction increases with every single 10 degrees Celsius. by 2 to 3%, 2 to 3%, 2 to 3 times, 2 to 3 times throughout. It's otherwise known as the temperature coefficient, and usually enzyme reactions are going to follow that because they are chemical reactions and you can apply it to most chemical reactions. But then the Q10, once you reach this optimal point over here, no longer applies, because after that you're inactivating your enzymes through denaturation. So it kind of just explains the slope over here and how much at each temperature increase are you going to expect your active or your reaction velocity to also increase. So that's it for can you repeat, please? Okay, so as this temperature increases up until your optimal point, you're going to have something called Q10, which basically for chemical reactions, the number that they use is 2 to 3. So the rate of your reaction as every 10 degrees Celsius and temperature increases, you can expect the reaction rate to double or triple. And then that's what forms this curve over here, is that reaction rate doubling or tripling. And this is something that you can apply throughout all chemical reactions. But to be honest, different types of enzymes respond to different types of temperatures. So that's why, like, for general practice, you can't always use Q10. It can't be 4 because after 40, it's beyond the optimal point. After 40, what do you, if you could reword your question? I don't think I quite understood it. At around like 37 to 38 degrees Celsius, most enzymes in your body tend to begin denaturing. So it can't be 4 because after 40, it's beyond the optimal point. Okay, yeah. If you could rephrase it, and then maybe I'll understand it. Q10 can't equal to 4 because at that point it begins to yes, exactly. It couldn't, because at that point it would begin to denature. You would want it to just be up until 2 to 3. Okay, the second thing then is your pH. Changing the pH will alter the charge of the enzyme, which in turn will alter the protein solubility and overall shape. Changing the shape or charge of the active site will diminish its ability to bind to the substrate, and that would ruin up your enzyme's function. So enzymes tend to have an optimal pH, usually 6 to 8 is good, but different enzymes have different optimum pHs. So here, your pepsin optimum is around 2, but your trypsin optimum is around 7.4. So this is kind of like to generally describe that, okay, if you're at an optimal of 7, because most kind of high school textbooks will tell you if you're at an optimum of pH of 7, that's good. Anything below it denatures, anything above it denatures, but this is kind of a more accurate form because different types of enzymes just have different pHs that they work best at, and at a pH maybe higher than that, they would denature, or sometimes at a pH lower, they would denature. And this is just because the different hydrogen that's in like an acidic bond might interrupt with, pepsin, or might interact with trypsin, but pepsin can withstand that, and etc. And then that would cause the denaturation. All right, and then the third graph that we talk about is substrate concentration. So if this was a reaction without an enzyme, we would expect that as the substrate concentration increases, the reaction velocity would also increase in like a proportional manner. So here it says the rate of velocity of a reaction is the number of substrate molecules converted to product per unit time. The increase in velocity is proportional to the substrate concentration. So as you get more substrates into the mix, you can expect more reactions to occur. So this would just be common sense. But then when you include an enzyme, faster reactions occur until all of your enzymes are saturated with substrates, and then reactions are going to plateau. So reactions will continue to occur, but the rate of the reaction will no longer continue to increase, despite your substrate concentration may be increasing. This is because they reach something called the Vmax, which is seen over here. That's basically the maximum amount of velocity that you can reach with an enzymatic reaction because you only have so many enzymes. So increasing substrate concentration usually starts by increasing the activity of the corresponding enzyme until you reach your Vmax, where the reaction velocity then plateaus despite the continuous increases in substrate concentration. And again, it's because of this concept of the environment being fully saturated with substrates and all your enzymes are bound and all of them are reacting and they're working at full capacity, and then they just can't do any more reactions. Okay, I'm going to see the question. Does 2 correspond to 20, 3 to 30, and 4 to 40, and so on? It's more so like for chemical reactions. Okay, we'll go back to for the Q10. Okay. Here, let me open up his slide so I can show you guys better. Okay. Basically, all he's saying here is that you have the Q10, which is going to cause this increase for 2 to 3. So it's like doubling or tripling the number of the rate of the reaction. So try to focus on like just this part over here. It doesn't really like correspond to like, okay, like 2 corresponds to 23 to 30 and stuff like that. It's more so just saying that like the rate of the reaction doubles at 2 to 3. So you can expect this number of like 2 to 3 to be like across all chemical reactions. So you like stop at that number and you understand that every time that I increase a chemical reaction's temperature by 10 degrees Celsius, I can expect the rate of the reaction to double or triple. With every rise of 10 degrees Celsius. But then that concept of Q10 stops after the optimal temperature, and then it no longer is doubling or tripling. After that, it's just denaturing. So yeah, it's just the temperature coefficient with every 10 degree rise. And Q10, yeah. Does that explain it a little bit better? Like the numbers of like Q, okay. All right. Okay, I think, that was better to use the slide for that. Okay, and now coenzymes and cofactors really quickly. He didn't spend much time on this, so I won't either. Coenzymes basically are organic and cofactors are inorganic. That is the main difference you need to know between the two. And coenzymes and cofactors both are just these little pieces of either protein or non-protein molecules that are going to go and bind to your inactive enzyme, which at that point you call an apoenzyme if it's inactive without its coenzyme or cofactor. And then it just enables the substrate to bind a little bit better to the enzyme. So when you have a complex of an apoenzyme, it's basically just the inactive enzyme, then the coenzyme or cofactor goes and binds to it. And then you have the complex of the coenzyme or cofactor with your apoenzyme. Now it's an active enzyme, also otherwise known as a holoenzyme. And then your substrate can easily bind to it. So same thing written over here. But another important thing to know is that coenzymes or co-substrates, they're not permanent parts of the enzyme structure. So as you can see, like the apoenzyme, when it's floating around without your coenzyme. The coenzyme is similar to an enzyme in that it gets recycled. It fulfills its little function and then it goes and it dissociates, and then you can recycle it with another complex as well. And like we said, cofactors are going to be inorganic. These are things like phosphates and metals, and then your coenzymes are going to be organic molecules. So those are basically the two main differences between the two. Another thing is that coenzymes are going to very loosely bind to your apoenzyme, whereas your cofactors are going to be tightly bound because it's going to be a covalently bonded group. And that's all he said also from this slide as well. So in summary, the most important things to remember from this lecture is that enzymes have an active site to which specific substrates bind. Enzymes work by lowering the activation energy of reactions. There are 2 models of actions of enzyme activity. There are 6 main classes of enzymes, and the temperature, pH, substrate concentration, and the presence of coenzymes affect the rate of activity of enzymes. Can you repeat about the loosely and tightly bound? Yeah, sure. So coenzymes, loosely bound, and cofactors, tightly bound. Cofactors also include prosthetic groups. Okay. All right, we're gonna really quickly, I'm gonna have you guys just do these 2 questions just to test your knowledge. These are the questions from the midterm that Academic Aid prepared, the mock midterm. So if you guys can go ahead and write your answers in the chat. This one is, I'm going to give you guys a little hint. I know that when we explained this, remember what X stood for? X, remember there were keywords of X, like methyl, acyl, amino, and phosphate. So that's your little hint. So if you hear the word phosphate, then you're already thinking of a functional group. So phosphate is a functional group that is transferring from one substance to another. So with that being said, yeah, exactly. The answer would be E, because transferases transfer a functional group, X, otherwise known as phosphate in this specific situation, from one substance to another. And an example of a transferase very well is kinase. Another example is transaminase. So that's another thing. I mean, if you wanted to memorize that, you could, but I think it's a lot easier if you just kind of memorize the structure instead. All right. Now, here is the second question. I don't know why clinical scenarios, I don't know why you guys get clinical scenarios if the question at the end is just which one of the following statements best describes the relationship between an enzyme, substrate, and product. So I mean, if it were me, I wouldn't really read the first part, but if you guys want to, you can. But go ahead and let me know what you guys think. Which of the statements is the most true? Let's start going through it from the bottom, and let's start deciding if each of the answer choices are right or wrong. So E says significant product formation results in activation of the reaction. Okay, someone direct messaged me the correct answer, but we're going to hold off before we say it. So significant product formation results in activation of the reaction. That's saying that the product formation has something to do with the activation of the reaction. But when we were discussing the things that can affect enzymatic activity, we never said anything about the product going back and affecting it, right? We said stuff about temperature, like an optimal temperature, pH, substrates, coenzymes, cofactors, things like that. So I wouldn't say E. Another thing is D. At low substrate concentrations, none of the enzymes are found in the ES complex. ES complex is just saying enzyme substrate complex. That's not really true either, is it? Because when we look at the substrate concentration, even if we only have a little bit of substrates, you can still expect some enzymes to hold on to those substrates. So it wouldn't really ever be zero. The rate of the reaction might be low, but it wouldn't ever be zero. So I would take out D as well. At high substrate concentrations, substrate-substrate interactions interfere with enzyme activity. Again, when we looked at this graph, we didn't really ever say anything about one substrate interacting with another substrate, and that being a reason why the enzymatic reaction couldn't continue. And then all the active sites of the enzyme are saturated with substrate at high substrate concentrations. So this is a high substrate concentration over here, and all of the rate of reaction is plateauing. So that would make sense, I think, if all of the active sites of the enzyme are saturated with substrates at high substrate concentrations. And then A says that enzyme product complexes enhance substrate binding. Again, we never really said much about enzyme product complexes, especially because enzyme product complexes, like seen here, are so short-lived. They dissociate, and then the enzyme goes back to another substrate, and then the product just goes off somewhere else. So through the process of elimination, I would say, and some of you guys have also said, that B is the most correct answer. This is because the rate of an enzyme catalyzed reaction generally increases exponentially with respect to the substrate concentration until the substrate concentration exhausts all of the enzyme active sites. And then once that occurs, you can expect the rate of reaction to plateau and be the same, regardless of the increase in substrates or whatnot, because all the enzymes have reached something called their Vmax, which is right over here, the velocity once it reaches its maximum velocity. And as the substrate concentration increases, we still don't have any more new reactions because everything is, all the enzymes are substrated. And so you can't increase the velocity of the reaction anymore. And then to write out the reasons why the other reactions or the other answer choices are not correct. Substrates cannot bind to enzyme product complexes, so that wouldn't be it. Because the substrate binding sites are occupied. Substrate-substrate interactions, which is answer number C, are the same regardless of the concentration of the substrate, and such interactions also don't affect enzymatic activity. And then the enzyme substrate complex, like we see in answer choice D, it can form at low substrate concentrations as well as high substrate concentrations. But again, you would never expect it to be 0. And then E is wrong because product formation does not stimulate enzyme activity and can slow down the reaction rate. So it doesn't really have much to do. If anything, you can expect it to slow it down because of the negative feedback in that situation. And yeah, with that being said, that concludes the PAL session for enzymology number 1. If you guys have any more questions, I'll definitely take them now. Oh, and this is my email. If you guys ever want to email me, if you guys want to send me a message on WhatsApp, you can take a picture of the slide. I'm more than happy to re-explain any of these different things. And also, if you guys just have any questions in general about first year or second year, whatever year you guys are currently in right now, because I know sometimes second years can take MOL 1 to 5 as well. You guys are more than welcome to message me whenever, and I'm more than happy to give advice. I have a younger sister who recently just finished first year, so I'm happy to give you guys the same advice I would give my younger sibling. So anytime. Yeah, you guys are welcome, no problem. Sorry,